Today's ALD Connect webinar series will be about clinical trials for ALD. My name is Stefan Kemp. I'm the principal investigator of the ALD group at the Academic Medical Center of the University of Amsterdam in the Netherlands. Before we start, I would like to read out a disclaimer. The information and services provided by ALD Connect are for informational purposes only. The information and services are not intended to be substitutes for professional medical advice, diagnosis, or treatment. If you or a family member are ill, or suspect that you or a family member are ill, seek professional medical attention immediately. ALD Connect does not recommend or endorse any specific physicians, treatments, procedures, or products, even though they may be mentioned on this site. Please note that your participation in this webinar is completely voluntary and that this webinar will be recorded and may be available for rebroadcast at a later time. So what are we going to talk about today? So I'm going to explain you about clinical trials, how they work, why we do clinical trials, and how we organize them. And then we'll move to more ALD-specific topics. We're going to talk about Lorenzo's oil, Lovastatin, Bezafibrate, and the trial that we did with women with ALD. And at the end, there should be time for questions and answers. But before I start with the webinar, I would like to introduce very shortly the AMC-ALD group. It's a group of dedicated physicians and researchers. The AMC has over 30 years of experience with ALD, and the strength of our research lies in the close interaction between the clinicians that are involved in the care of ALD patients and the researchers. This enables us to translate new scientific findings rapidly to the clinic, and vice versa, translate clinical questions into their scientific components. The principal investigators of the AMC-ALD group are Mark Engelen, here at the top right, and myself. So why do we need clinical trials? Well, technically, or basically, we need clinical trials to test whether a particular treatment is safe and beneficial for human use. And to answer two main questions. Does a treatment work? So that deals with efficacy. And are there any side effects? That deals with safety. Clinical trials help us to take the guesswork out of healthcare. And to answer the question, does the treatment really work? And it's very important because you have all heard about the placebo effect. And I would like to spend 2 slides, well, a slide and a short movie on the placebo effect. It's commonly said that the placebo effect is the effect of a treatment such as a sugar pill. But in fact, it's the effect of a therapeutic encounter, which is everything that surrounds that sugar pill, which includes, but is not limited to, the provision of care, the clinical context, the medical ritual, the trust, compassion, and hope. It's the effect that is due to the meaning that the treatment has for the patient. And placebo, there are very strange things with placebo effects. I would like to spend 2 minutes on a short movie. So here we go. I don't hope it's too loud. Oh, sorry. Oh.
Now you've all heard of the placebo effect before, when something with no known therapeutic value can actually make people feel better. It's a great trick our minds play on us, that by believing and expecting something to work, it actually does. But what's weird is that the strength of the effect can differ for some really strange reasons. For example, the same placebo can treat pain half as well as aspirin, while at the same time treating pain half as well as morphine. Morphine's a much more powerful painkiller, but a placebo is half as effective as both. Saying a placebo will reduce pain reduces pain, but saying that the same placebo will increase pain increases pain. Believing that a placebo will make you feel better will make you feel better. Believing that it won't has the opposite effect. Now, placebos aren't just pills. They can be creams, injections, surgeries, or drinks. You can even get placebo buttons. They don't actually do anything, but they sure as hell make you feel like you're in control. But not all placebos are equal. The effect of the placebo is bigger when the pill itself is bigger, or if you have 2 instead of 1, or 2 once a day instead of 1 twice a day. And a capsule will usually beat a pill, and a syringe will usually beat a capsule, and anything with a big-ass science machine can outperform any of them. A plain pill works worse than a branded one. A discounted pill works worse than a pricey one, and even a pill in a plain box does worse than one that's all shiny and shit. Placebos that are blue work best as downers, and placebos that are red are better as uppers. Studies have shown that people who take their meds on a regular basis are less likely to die than those that don't, even if those meds are all placebos. You can even get addicted to placebos. In one study, a group of women took placebos for more than 5 years. 40% of them suffered withdrawals afterwards. In fact, the effect of placebos can be so strong that some people want them banned from sports. But I mean, how would you even test for that? Placebos don't even seem to work from place to place. For example, in Germany, using a placebo to treat ulcers works better than anywhere else in Europe. But using a placebo that treats hypertension doesn't work nearly as well as it does for its neighbours. Now, remember that all of this is about comparing things that both have nothing medically effective in them, which goes to show that a placebo isn't about what's in it, but about the beliefs that we load onto it. Our minds create the medicine, and that is pretty freaking weird.
Okay, so the oldest record of a clinical trial, so that's why we need clinical trials. The oldest record of a clinical trial dates from the Old Testament, from the book of Daniel. It describes how King Nebuchadnezzar II, the king of Babylon, ordered his people to eat only wine and drink water, only meat and drink wine, because he believed that this diet would keep his people in perpetual good spirit. However, some young men of royal blood requested to follow a diet of vegetables and water. The king, his curiosity triggered, permitted these young men to follow a diet of vegetables and water, but only for 10 days, after which he would assess their health. The results of this trial showed that the young men were noticeably healthier and more energetic than the people of wine and the meat diet, and the king allowed the royals to continue the diet. So we talk about clinical trials. You will hear all these, read about all this terminology, and I would like to spend 2 slides on explaining some of the most commonly used terms. So there's an alphabetical order. So an adverse event is an unfavorable change in the health of participants. An arm or a treatment arm is a subgroup of patients that receives the intervention, so the medication, or no intervention, hence the placebo. Baseline characteristics is the data that is collected at the beginning of a study for all participants, like age, gender, and study-specific measurements. For ALD, we would, for example, measure varying chain fatty acids or the walking distance a patient can walk. Blinding or masking means that participants and researchers do not know who gets which treatment at what part of the trial. Exclusion criteria are the factors or reasons that prevent a person from participating in a clinical study. Inclusion criteria are the factors and reasons that allow a person to participate in a clinical study. An informed consent is a document that explains how the study works as well as its specific risks and benefits. Patients can still leave the study, even when the study has started. The IRB, the Institutional Review Board, is a group of physicians, researchers, and members of the community, which are often patients from patient organizations, who ensure that clinical trials are ethical and that participants' rights are protected. An observational study is a study without medication. An open label study is a study that involves no blinding, so everybody knows who gets what at which moment of the trial. Outcome measurements or endpoints are planned measurements that are described at the beginning in the study protocol that used to determine the effect of the interventions of the participants in the clinical trial. And these are defined beforehand. A placebo, well, we heard about placebos, it looks and tastes like a real pill, but there's no medication. And randomization, finally, participants as well as the investigators do not choose which participant is in the control group or arm, or which are in the experimental group or arm. The participants are assigned by chance, often by a computer. This is done to avoid bias. So a clinical trial is is pretty lengthy, and it's shown on this slide. Before any medication can be tested in a human being, there's the preclinical period. And that can, well, it says here 6.5 years on average, but it can be much longer. And once a drug reaches the clinic, there are 3 different phases, clinical phase, trial phases. So we have phase 1, phase 2, and phase 3. In phase 1, researchers test a new drug or treatment in a small group of people for the first time to elevate its safety, determine the safe dosage range, and identify side effects. Then we move to phase 2. There's a few or more patients. In phase 2, the drug or the treatment is given to a large group of people to see if it's effective and to further evaluate safety. And then we go to phase 3. In phase 3, the drug or treatment is given to a large groups of people to confirm its effectiveness, monitor its side effects, and compare it to the commonly used treatments, and collect information that will allow the drug or treatment to be used safely. If the drug survives all these 3 phases, we talk about it, then we go to FDA approval. And when the drug is marketed, we go to phase 4. And in phase 4, studies are done after the drug is or treatment has been marketed to gather information on the drug's effect in various populations and any side effects associated with long-term use of the drug. So you see high numbers of patients here. But if you talk about rare diseases, these are patient numbers that we can never reach. So for rare diseases, the number of patients in each of the 3 phases will be lower. But the investigations will be the same in each of the phases. So why does it take so long to find an effective drug for any disease? Well, there are many answers to this question. Just to name a few. First of all, we need to understand the disease. The genes that are affected, the enzymes, the metabolites, the organs that are involved. Then we have to find a therapeutic target. Next, a drug that can affect the therapeutic target in any beneficial way. But the drug needs to be effective, not just in the preclinical stage, for example, in cell lines of patients or in a mouse model, but it also needs to be effective in patients. And of course, it has to reach its target tissue. And there are many additional reasons why a drug might fail. And this is clearly exemplified in this image. The average time needed to go from the drug discovery through the FDA approval is at least 10 years on average, and the cost involved in finding an FDA-approved medicine is about $2.6 billion. It's been calculated that from the drugs that reach phase 1 studies, less than 12% eventually is being approved by the FDA. Okay, this is a very basic clinical trial design. So we start with a group of patients here on the left. They are, by randomization, they go into one of the 2 treatment arms. So here we have the group that receives the intervention, so the medication, and the other group will receive is the control group and will receive the placebo. And then after the intervention, the clinical endpoints, which were defined before the study started, will be measured, and the 2 groups will be compared. And now we have an answer whether our new medicine is effective, yes or no. So why do we need clinical trials for ALD? Well, we do not need a clinical trial to test the efficacy of bone marrow transplantation in boys affected by cerebral ALD. Untreated cerebral ALD is progressive and it's often fatal, which is shown here in this Kaplan-Meier graph. This plot also shows the excellent survival outcomes of which could be achieved in boys transplanted at early stages of cerebral ALD, defined as 0 to 1 neurological deficits and MRI severity scored less than 9. The survival of these boys, transplanted boys, was 92% after 5 years, versus 30 to 40% of the all untransplanted boys. So how about Lorenzo's oil? Lorenzo's oil was developed about 30 years ago, and after 30 years we still do not know whether Lorenzo's oil is beneficial for ALD patients. And this deserves a little bit more explanation, which I will do in the next 10 slides. But before we talk about Lorenzo's oil, we first have to understand the biochemistry of ALD, and that is shown in this graph. So this is a typical diagnostic graph in which we measure the C26, which is the very long chain fatty acids, or abbreviated as VLCFA, and they're shown here at the bottom. This is a C26 fatty acid with the polar head group and the long fatty tail, consisting in total of 26 carbons. And if we measure these fatty acids in plasma from patients with ALD, and we compare it to controls, so here we have the controls in green, so in this gray bar here is the control level, we see that all males have elevated levels of very long chain fatty acids in their plasma. And about 85% of the females have elevated levels of very long chain fatty acids. This also means that if a female is suspected to have ALD, a measurement of very long chain fatty acids that is normal does not exclude ALD. And then other ways of diagnosis must be applied, like genomic sequencing. So this is a very simple cell with the nucleus where the genetic material is located. We recognize the mitochondrion here and the peroxisome. And here are the very long chain fatty acids. So here they're sitting in the cell. And these fatty acids, they are generated, or they originate not so much from the diet, but they're produced in the cell. And I will show that in a few slides in a little bit more detail. So the very long chain fatty acids, they originate from elongation or synthesis from long chain fatty acids. And there's a tight balance between the production of very long chain fatty acids and their breakdown. For the breakdown of very long chain fatty acids, we have this protein here, ABCD1, which is the ALD protein. And the ALD protein transports the very long chain fatty acids from the cytosol in the cell over the membrane of the peroxisome, where they are degraded or broken down. As you can see, there's a tight balance. In ALD, there's a genetic defect in the ABCD1 gene, which causes a malfunctioning of the ABCD1 protein, and thereby there is less transport of very long chain fatty acids from the cell into this organelle, this only organelle where they can be degraded. So the breakdown goes down, the production remains more or less at the same rate, but the balance is, there's a disbalance between the production of fatty acids and the breakdown. And this results in an increase in very long chain fatty acids and the storage of very long chain fatty acids. And that brings us back to this graph. So here you see the buildup of C26 levels. This is for plasma, but you can this is the same for blood cells. It's the same for brain, spinal cord, myelin, kidneys, liver, any tissue. And eventually, this causes the disease. So here we see these strange white needle-like structures. They were already recognized in the 70s by Jim Powers and Schomburg. And these are very harmful. Okay, so back to the biochemistry. So I told you that very long chain fatty acids, they're synthesized from long chain fatty acids. So this C16, that's a typical long chain fatty acid. Interestingly, this C16 and C18, these fatty acids, can also be desaturated. And what happens is that a double bond is introduced in the fatty acid chain. You can see here. So this is a saturated fatty acid. It's a straight chain fatty acid. And if there's one double bond, there's a nick in the side chain. And these unsaturated fatty acids have very different chemical and physiological characteristics than saturated fatty acids. And these fatty acids, these green arrows indicate that there is elongation of the fatty acid side chain. And it means that there are just 2 carbon atoms are added to the side chain. So you go from 16 carbons to 18, to 20, to 22, 24, and 26. And the problem in ALD is with the C26. In 1986, Dr. Bill Rizzo and Paul Watkins from Kennedy Krieger and University of Virginia demonstrated that when you add unsaturated fatty acids like oleic acid and uric acid to tissue culture, so these are little flasks in which skin fibroblasts, skin cells from ALD patients are grown, and this red stuff is the liquid, is the tissue culture medium. And when you add oleic acid and uric acid to this tissue culture medium, that you can observe a decrease in the C26 levels inside these cells. And the reduction can be as strong that the levels become completely normal. So the researchers hypothesized that the enzymes that are responsible for the elongation of these fatty acids, so the chain lengthening of these fatty acids, are the same for the monounsaturated or the unsaturated fatty acids and the saturated fatty acids. But that our affinity for the monounsaturated fatty acids is higher than for the saturated fatty acids. So there's competition between these fatty acids and these fatty acids. So if you fill the system like they did in the tissue culture experiments with oleic acid and uric acid, so the unsaturated fatty acids, the cell generates less of the C26 fatty acids because of competition. And we will go into this a little later in the webinar. So soon after these findings, an oil is generated, and the oil is referred to as Lorenzo oil, which I will show you on the next slide. And Lorenzo oil is a mixture of oleic acid, so the C18-1, and uric acid. And the parents of Lorenzo odone played an important role in this, and therefore the oil is referred to as Lorenzo's oil. So when ALD patients start using the oil, the plasma very long chain fatty acids, again here, so here we have the elevated C26 levels, and this is the weeks of treatment. The plasma very long chain fatty acids normalized within a few months. And they remained in the normal level. So these results were published, and in 1987 in the Annals of Neurology, and the abstract is very clear in its recommendation. So let's go through this abstract. So it says we can read that the treatments of patients with the Lorenzo's oil resulted in a reduction in the levels of plasma C26. But not in all patients, in 25 out of 36 patients. And that 2 patients out of these 36 patients showed signs of clinical improvement. On the other hand, 2 patients worsened while they were on Lorenzo oil. And there's a clear recommendation in the abstract. The authors say that a clinical trial is indicated to test whether the approach, whether Lorenzo's oil is beneficial for ALD patients. Unfortunately, this clinical trial was never done. So what happened is that all patients received Lorenzo's oil, were starting to take Lorenzo's oil, and no patients, so there was no study in which a placebo was included. But within a few years, publications appeared by the group of Patrick Aubourg in Paris, in France, and Björn van Geel from the AMC in Amsterdam, that demonstrated that Lorenzo's oil does not protect patients against development of clinical symptoms. Furthermore, patients that use the oil develop cerebral ALD and or AMN despite they were taking the Lorenzo's oil. So in 2005, a paper was published by Hugo Moser and colleagues, that published the experience in a large group of boys who had normal MRI at their entry into the study, and in this group, very long chain fatty acids were lowered by Lorenzo oil, and the relationship could be demonstrated in a reduction of the risk of developing childhood cerebral ALD and the reduction of the very long chain fatty acids. So while these boys were using Lorenzo oil, still 24% developed MRI changes. So is this a positive effect? The problem is that nobody exactly knows the real percentage of boys that develop cerebral ALD. The literature says that this figure is 37%. But this figure is based on historical controls, and it's not based on a placebo arm in this trial. And there are arguments that there's a very good chance that many patients, especially the mildly affected patients, were missed in the figures that ended up in the calculation that 37% of untreated boys develop childhood cerebral ALD. So therefore, the 37% may be an overestimation. Nobody knows what the exact percentage is. The newborn screening is implemented in New York State and from next year also in the Netherlands. We will finally know this answer, but that will take another 10 years. So what if the real percentage is not 37%, but it's smaller? It's smaller than 24%. Then we have to conclude that Lorenzo oil did not work in this trial, and that may possibly even be bad. If the percentage of boys that develop childhood cerebral ALD is higher than 24%, then we have to conclude that Lorenzo oil protected a part of the boys, but not all of them, because still 24% of the boys develop childhood cerebral ALD. So the only way to find out and to enter this uncertainty is a placebo-controlled clinical trial. And this was fully recognized by the people at the Kennedy Krieger Institute in Baltimore. So in 2007, they started a phase 3 placebo-controlled trial in Baltimore. The trial would last for 4 years, and it would include 240 patients, 120 men with AMN without cerebral involvement, and 120 women with ALD. There were two problems with the trial. Some patients that refused to participate in this trial because they had a 50% chance to end up in the placebo group, or some of them stopped when they found out that they were in the placebo group because they had a very long chain fatty acid measured somewhere, and when these were not reduced, they knew that they were in the placebo group and they stopped participating in the trial. But there was a second problem. The study was eventually aborted before completion by the Safety Monitoring Board because of presumed side effects of the placebo treatment. Placebo treatment. Yes, so the study was aborted. So to conclude, does Lorenzo oil have any clinical effect? After 30 years, we still do not know the answer to this question. What we do know is that Lorenzo oil, we have to conclude that Lorenzo oil has never been properly investigated. While it normalizes plasma C26 levels, the effect on very long chain fatty acids in tissues, like brain, spinal cord, is unknown and unreported. In countries like the Netherlands, in France, patients, individual patients may take Lorenzo oil, but in general, patients do not take Lorenzo oil anymore. And unfortunately, the use of Lorenzo oil in patients may even hinder new clinical research. For example, the use of Lorenzo oil is often an exclusion criteria in clinical trials, as we will see in the next slides that I will show you. Okay, so that was Lorenzo oil. Now we move to another intervention, Lovastatin. In 1999, or 1998, as it says here, the New England Journal of Medicine published that cholesterol-lowering drug Lovastatin reduced very long chain fatty acids in ALD. And if the New England Journal of Medicine publishes a paper like this with these type of results, this may be considered the therapy. And indeed, if you look at the table, here is the fatty acid levels before treatment, and here after treatment, and we see a nice reduction over a period of 6 months. However, among ALD researchers, there was quite some debate about this paper. For example, the study lacked the analysis in healthy controls, because if you can see, there are 7 patients here involved, but there is no value mentioned for controls. Furthermore, what was reported is plasma very long chain fatty acids. What does this mean? What are plasma very long chain fatty acids? It sounds like a silly question, but it's not, and I will explain you on the next slide. Plasma very long chain fatty acids is the sum of those fatty acids that are considered very long chain fatty acids. And those are C22, C24, and C26. So it's the sum of these 3 fatty acids. Importantly, it's C26 that's elevated in ALD. And more importantly, if you calculate the ratio of C22 to C24 to C26, it's about 100 to 100 to 1, as shown here. This is a typical analysis of very long chain fatty acids coming from our laboratory. So if we do a simple math and we add 22 micromolar plus 61 micromolar plus 1, the total is 134 micromolar of very long chain fatty acids. So what if we have any intervention study and the intervention would only affect the concentration of C22, let's say by 50%. Then the 72 will be reduced to 36. This will remain the same, so 36 plus 61 plus 1 is 98. So this is a nice reduction in the sum of plasma very long chain fatty acids. However, as you can see, in this hypothetical model, for this hypothetical drug, there is no effect whatsoever on C26. So while this reduction is shown, it's rather meaningless because it doesn't tell us anything about the effect on C26. Let's see. So the question is, what's the true effect of Lovastatin or any other cholesterol-lowering drug, statins, on the C26 levels? Another important thing is that in plasma, very long chain fatty acids are not swimming around freely. They're bound to LDL and HDL cholesterol. It's shown here. This is an LDL particle, and here are cholesterol esters, and it means that there are fatty acids attached to it. These are triglycerides. There are also fatty acids attached to it. These are phospholipids. They also contain fatty acids, and these fatty acids that are attached to cholesterol, triglycerides, and phospholipids can be very long chain fatty acids. So we hypothesized that a decrease in the total cholesterol in plasma would correlate with a reduction in the sum of all very long chain fatty acids, simply because there would be less binding spots in the plasma. So the only way to find out whether our hypothesis was true or whether the publication in the New England Journal of Medicine was true is to perform a randomized, double-blind, placebo-controlled crossover trial. And I will explain you what we did. And the question that we wanted to answer is, does Lovastatin have an effect on plasma and blood cell C26 levels in ALD patients? So what we did, these are the inclusion criteria. These are males. For this study, males could participate. The age should be older than 18 years, and they should have AMN, the AMN phenotype. Exclusion criteria are, of course, the use of cholesterol-lowering drugs and Lorenzo's oil, because the question that we want to answer, does the treatment have any effect on C26 levels? I've just shown you that Lorenzo's oil is perfectly capable of lowering C26 levels. So that will be clearly an exclusion criteria. We calculated that by statistics that we would have at least 10 patients needed in this trial, and in the end 14 patients participated in the trial. So they started all on a standardized diet from the American Heart Association. And then by computer and by chance they were divided in 2 groups. One group received the treatment, in this case 40 milligrams of placebo of Lovastatin, and the other group would receive placebo. But nobody knew who was receiving what at any stage in the trial. And this is a very powerful trial design because they would receive, for 3 months, they would receive the intervention, and then there will be 1 month without treatment, and then the patients switch treatment arm. So the placebo group would go to Lovastatin, and the Lovastatin group would go to placebo. And then for another 3 months, they receive the medication, and then there will be the end of the trial. And blood was drawn at the end of the low-fat diet, the beginning of the first treatment, and obviously, and in the middle and at the end, the beginning of the second treatment arm, the middle and at the end, and the end of the trial. So here are the results for the Lovastatin trial. Well, Lovastatin is a statin which people take to lower their cholesterol, or the LDL. And even though LDL cholesterol is not elevated in ALD, the patients still lower their LDL cholesterol while using Lovastatin. The plasma C26 levels did reduce. So here we get the real effect of Lovastatin, which is only like it's statistically significant, but it's a reduction of about 20%, and it's far from normalized. Normal levels would be well below 1, around 0.5. More importantly, what is the effect of Lovastatin in blood cells? So here we look at the effect of treatment on active metabolism, so active synthesis and active breakdown of C26. And as you can see, the placebo group, the green group with the Lovastatin treatment group, there is no effect whatsoever. And we hypothesized that the effect would be lowering the amount of LDL particles and thereby lowering the amount of binding spots in blood for C26. And that's shown here because if we isolated, when we isolated LDL particles and then specifically measured in these cholesterol particles the amount of C26, there was no change. So to conclude, yes, Lovastatin does lower plasma C26 levels in patients with ALD by roughly 20 to 30%, but this is absolutely not a normalization. Furthermore, and more importantly, there was no effect of treatment on C26 levels in blood cells, and this can be considered a measurement or a measure of tissue C26 accumulation. The C26 reduction in plasma can be considered an epiphenomenon of the reduction of LDL cholesterol because C26 is present attached to these LDL particles. The off-label use of Lovastatin for ALD and AMM patients should be discontinued, and the conclusion is that Lovastatin is not a therapy for ALD. Let's move to Bezafibrate. Bezafibrate, I will introduce that as well. It also has to do with the synthesis of very long-chain fatty acids. Okay, so I just told you that the C26 levels that are elevated in ALD are generated by elongation of long-chain fatty acids. So they go from C16, C18, 20, 22, 24, and 26. And these are enzymatic reactions. And these enzymes are called elongators that put the 2 carbon atoms to the side chain. About 10 years ago, we set out to identify the enzymes that are responsible or carrying out these elongation steps. And our results demonstrated that the enzyme that's important for the elongation of C16 to C20 is EloVL6 or elongate 6. Then another enzyme takes over. It's called EloVL1 or elongate 1, and that elongates C22 to C24 to C26. We hypothesized that because there is now a specific enzyme that generates the C26, that thereby EloVL1 could be considered as a therapeutic target, and we hypothesized that an inhibition of this enzyme, EloVL1 or elongate 1, would result in a lower production of C26 and most likely lower levels of C26. Okay, now I skip about 5 to 7 years of research, because the theory is nice that you can set up a hypothesis that you can inhibit ELO-VL1, and there are ways to demonstrate that. But if you want to do that, demonstrate it in a mouse model or in a cell line or in a patient, you need to have a drug that inhibits ELO-VL1. So, like I said, that took about 5 to 7 years, and then we stumbled upon Bezafibrate, and here is the key experiment. We isolated the ELO-VL1 enzyme, and here this graph shows the activity of the enzyme. So if you don't add any bezafibrate, this is the activity. You have 18 units, and if we add bezafibrate, where's my mouse? If we add increasing concentration of bezafibrate, the activity of ELO-VL1 is inhibited up to 80%. Then the question is, does this reduced activity of ELO-VL1 also have an effect on C26 levels? And that's shown in this graph. Here we measure the synthesis of C26. And if we do not add bezafibrate to ALD cells, and these are ALD cells, the blue ones, this is the amount that the C26 that these cells produce. If we add bezafibrate in increasing concentrations, up to 400 micromolar, and then you can see that with 400 micromolar of C26, bezafibrate added to the ALD cells, these ALD cells produce the same amount of C26 as control cells do. The good thing about bezafibrate is that bezafibrate has been used in the clinic for more than 30 years. And we talked about these studies at meetings, like at the ALA meeting and at the ULF meeting. And because bezafibrate is an over-the-counter available drug, we were approached by patients. And they said, well, I really recall one gentleman, he came to me and he said, well, I cannot buy bezafibrate in the US, but if I take my car and I drive across the border, I can go to Canada and I can buy bezafibrate. When should I start? Well, I urged this person to not start using bezafibrate, but wait until we would finish the clinical trial. And we made a promise that we would finish the clinical trial within a year after that presentation. So what we did an open label biochemical trial. So I talked about placebos, but in this case we did not use a placebo, and I will explain why we did that. One of the main reasons is time. Placebo-controlled trial takes much more time because you need to develop your placebo and also money. The bizarre thing is that with the Lovastatin trial, the drugs cost about $1,000, but the generation of the placebos was about 10 times more expensive. So in this case, we went for an open-label clinical trial. And the trial was funded by the Stop ALD Foundation. We included 10 ALD men with the AMN phenotype, and again the exclusion criteria were cholesterol-lowering medication and Lorenzo's oil. The design of the study was a dosage escalation study. Patients would receive 3 months of bezafibrate, and then the drug concentration will be increased to 800 milligrams. And the study would go on for another 6 months. And the design was such a way that each patient is its own control. And that's why we could do an open label trial, because the baseline measurement would be the C26 level in blood cells at baseline, so here at visit 0. And this measurement we could compare with the C26 levels at the end of the 400 milligram trial, and with the C26 levels at the end of the 6-month treatment. So here are the results. Well, bezafibrate is a drug that's very effective in lowering triglyceride levels in patients that have problems with triglyceride levels. So as for Lovastatin, ALD patients do not have problems with triglycerides. But even using 400 and 800 milligrams of bezafibrate still lowered the triglyceride levels in ALD patients. And here are the results for the C26 levels in plasma and in blood cells. Here in green is baseline. here is the 12 months, so after 3 months and after 6 months, and this is the levels on control. So as you can see, unfortunately, bezafibrate did not have any effect neither in plasma C26 levels nor on, more importantly, on blood cell C26 levels. So this is a very disappointing result, but luckily we have an explanation for a very likely explanation for these results. Why was bezafibrate ineffective? Well, we had the blood collected from the patients, and we used the blood to measure ferro longevity acids, but we also set up an assay to measure the bezafibrate concentrations. And just after a few hours after taking the medication, when we measured the C26, the bezafibrate levels, they did not exceed 25 micromole per liter. And you may recall from a few slides before that in ALD cells we had to treat these cells with 400 micromolar of bezafibrate to get a normalization of C26. So this is incorrect, but if we take this 25 micromolar and we just add it to the graph, then we're sitting here. And this is comparing apples and oranges, I know, but if we just do that, why not? Then we see that, yes, knowing after the trial that we only could reach 25 micromolar, this may be an explanation why bezafibrate was ineffective in patients. However, ELO301 is still a therapeutic target because our preclinical studies clearly show that ELO301, if you reduce ELO301 by any means, that it directly affects the amount. But in order to inhibit ELO301, we need a more potent compound. So either we need to find another drug that inhibits the enzyme, or we need to find ways to either modify bezafibrate to make it more effective, or to maybe include it in nanoparticles or whatever. So this is all research that's still going on. But over-the-counter bezafibrate is not a therapy for ALD. So when we move to the last part of the presentation, and that's a study that involves women with ALD. So for many years, let's say, how do I start this? Well, the signs and the clinical symptoms that women with ALD, which were previously referred to as ALD carriers, well, we now call them women with ALD, because women with carriers implicates that they do not have clinical signs, and that's untrue, which I will show you. So the signs and clinical symptoms that women with ALD develop had never been properly reported or investigated. And to investigate the symptoms and their frequency in women with ALD, we set out to do a study. And with the aim to generate an unbiased group, as unbiased as possible, group of women with ALD, my colleague, Dr. Mark Engelen, invited women with ALD that visited the outpatient clinic, either for themselves, for yearly checkups, or because they accompanied their affected son or any other family member, to participate in the clinical study. To further reduce the amount of bias, these women were also asked to spread the word in their family and to see if they had cousins or sisters that would participate. So we, in the end, a total of 46 women participated in this trial. And the trial was carried out between 2010 and 2012, and the results were published in 2014. And we plan to do a follow-up study either next year or the year after to have an idea of the natural history in women with ALD. So this is one of the most important results from the trial. This shows that the frequency of symptomatic women increases sharply with age. So here we see these little black dots here are the individual women, and here it simply says no, symptomatic, no or yes. and they're plotted. And you can see that the majority of the no's are sitting at the young age, and with increasing age, all women develop symptoms. And if we put it as a group, we see that the age group between 20 and 39 years of age, 18% of women had were symptomatic, and in the 40 to 59 group, this increased to 80%. And in the older group, from 60 to 79, it increased to almost 90%. So this is a table out of the paper. Dr. Engelen did extensive analysis with an emphasis on neurological complaints and neurological examination. In particular, he investigated the symptoms of incontinence, gait disorder, maximum walking distance, and sensory disturbances were recorded. And the table presents the results ordered by the 3 different age groups. And we see that almost all of these analyses revealed that their incidence increased with age, up to 90% for pathological reflexes myelopathy, which is spinal cord disease, and being symptomatic. The conclusions are that ALD carriers develop signs. Sorry, I say ALD carriers, it should be women with ALD, develop signs and symptoms of myelopathy, which is spinal cord disease, related disease, and or peripheral neuropathy. And that women with ALD are highly likely to develop symptoms. And the most important predictor is age. So if they become older, the chances to develop symptoms increases. That brings me to my conclusion slide. Clinical trials are needed to answer whether a drug or a therapy is truly effective and safe for human use. I talked about Lorenzo's oil, and unfortunately we have to conclude that Lorenzo oil has never been properly investigated for different reasons. There's no scientific evidence to prove that Lorenzo's oil is a disease-modifying on AMN, I showed you in the published papers. Lovastatin is not a therapy for ALD, Bezafibrate is not a therapy for ALD, and women with ALD are highly likely to develop symptoms. And the most important predictor is age. And I forgot to say that the paper by Dr. Engelen that was published in Brain is available through ALD Connect. I just sent it to Ina, and if you use the email address that's presented at the end of the webinar, and you are interested in this paper, you can send her an email and you can request the paper. Well, while you read the disclaimer, I'll take a sip of water, and then I have one more slide. I would like to thank you for your attention, and I have no idea what the time is. Yeah, we have a few more minutes to answer questions, and I'm happy to do that. Now it's going to be tricky because now I need to find those questions. I have a question here. The question is, does Lorenzo's oil lead to an increase in C20 monounsaturated, C26 monounsaturated, etc., etc.? That's a very good question. Yes, it does. Why did I decide not to include that in the presentation? Yes, we let's think in 2005 we published a paper in which we demonstrated that the synthesis of very long chain fatty acids is increased in ALD patients. And what we did, we had plasma stored from patients that had been using Lorenzo's oil, and those were stored in our -80 freezers. And retrospectively, we collected patient plasma from, if I remember correctly, three patients at baseline and up to 1.5 years into the treatment. And what we demonstrated that yes, indeed, the C26 levels were reduced. Actually, that was the graph that I used in this webinar. But if you indeed look at the concentrations of C18-1 So the monounsaturated fatty acids 18, C18-1, C21, C22-1, C24-1, C26-1, C28-1, C31, and I think we also did C32-1, which is the really long fatty acids, that all of these increased. So the question is, yes, Lorenzo's oil leads to an increase in the monounsaturated fatty acids. The question that's not asked, but the question that was related to this, is that toxic? That we do not know. We do not know whether these monounsaturated fatty acids are toxic. We do know that the C18 and C22-1 is not toxic, but these extreme long fatty acids we do not know. Then there's another question. That's a good question. I mixed it up myself, I noticed. The question 2 is, when you say women with ALD, are you referring to carriers? Yes. In the beginning, until a few years ago, well, in general, women with ALD, proven ALD, so mutation in the ABCD1 gene, are referred to as carriers of ALD. The problem is that, and it's more semantic, is that if you talk about a carrier, people who are unfamiliar with the disease, and they know that it's an X chromosome linked disease, often think that, oh, it's X linked, you're a woman, you have your backup X chromosome, you probably won't develop any symptoms. And our study clearly demonstrated that that's not true. That's not true. So we try to use, instead of ALD carriers, use the word women with ALD. And women with ALD does not, by definition, means that they have clinical signs of ALD, but they have proven ALD, both at the genetic level, so they have a mutation in the ABCD1 gene, and they likely, 85% of them, likely have elevated variant chain fatty acid levels, and they can have ALD symptoms. So yeah, women with ALD are ALD carriers. There's another question. What about antioxidants? That's a very good question. It's not a topic of this webinar, and I think that there's a fair chance that this will be subject of a webinar in the future. So I'll pass that on to the next speaker in any of the next webinars. There's another question. It does not seem that there are many studies which you shared that include young ALD boys, like ages 18 months to 10 years. Or are there but that you did not discuss today? I did discuss. I did not discuss about boys, young boys, in relation to Lovastatin or Bezafibrate. Those were trials that were only done with men with AMN, but the bone marrow transplant studies are typically for boys, early boys with cerebral ALD. And the whole thing I talked about Lorenzo oil, the effect of treatment on the boys that developed childhood cerebral ALD, the 24%, that deals with boys with young boys in that age group between 2 years and 10 years of age. Why do we clinical trials in men? That has to do with informed consent and stuff like that. There's another question. Can the measure of C26 be a primary endpoint for ALD and AMN clinical trials? If so, how long does the dosing needs to be? It's a very good question. Well, there are clinical trials in which the endpoint is a clinical endpoint, like, for example, walking distance, or how much time it takes you to get out of a chair, walk a few meters and go back. It depends on the target. If the target of the treatment has to do with fatty acids, so like for Lovastatin, the target was very chain fatty acids with bezafibrate. With bezafibrate, is directly targeted against towards C26 because it targets the enzyme. Then the primary endpoint would be biochemical C26. There's also another reason. If the target is biochemical, we can get away with trials that take up to a few months. But since if the endpoint would be clinical, because the clinical development in individual patients is so unpredictable, these trials will take longer, and if it's not C26 as an endpoint, then it's a different target. What do I think about lowering plasma C26 in women with ALD, such as Lorenzo's oil? Well, the same as for men with AMN. The reason I say that that women do not develop AMN is because the abbreviation of AMN is adrenomyeloneuropathy, and the A stands for adrenal insufficiency, and women do not develop adrenal insufficiency, so we cannot say that women develop AMN, but they do develop a comparable spinal cord disease, a myelopathy, as men with AMN. So, yeah, Lorenzo's oil has been studied in men with AMN, and it's demonstrated not to be effective because it does not protect these men that take Lorenzo's oil for many years to develop clinical signs and symptoms of AMN. So the answer to this question would be the same. Would be that I don't expect a difference in outcome between women with ALD and men with AMN. And in fact, the trial that was started in 2007 at the Kennedy Krieger Institute addressed this issue because it would include 120 men with AMN phenotype without cerebral involvement and 120 women with ALD. So to answer a question that's not been asked, nowadays, most of the time trials include men with AMN, but from now on, it's also that many of the trials that are ongoing are hopefully will be planned in the near future. It will involve both men with AMN and women with ALD. I don't think we should make a difference between men and women. If there are no other questions, then I think, wait, there's another one. Does the level of viral chain fatty acids in women affect the chance to have symptoms? That's a very good question. That's one of the items that we investigated in the women with ALD paper that was published in Brain. We hypothesized that, how can I say this in a few sentences? We thought that yes, there would be a chance if the viral chain fatty acids are normal. So in 15% of women with ALD, so with a defect, they have normal levels of viral chain fatty acids, and it probably has to do with the fact that they have 2 X chromosomes. I probably need to spend another webinar on explaining that, but we hypothesized that there will be a chance that women with normal or lower levels of viral chain fatty acids may have less symptoms, but the limitation in these type of investigations are that we cannot measure viral chain fatty acid levels in the target tissues, and in this case, it would be the spinal cord. So we have to do with what we get and we can get, and those are blood cells and skin fibroblasts. And in skin fibroblasts, we could not demonstrate a correlation between those women that have symptoms or the severity of the symptoms and the levels of viral chain fatty acids. But it's still an open question. my hypothesis would be that there should be such a correlation, but it may take a long time before we can answer that question, and it's basically due to limitations of what we can measure and where we can measure in a patient. I see a new question popping up. Provision so that we know. This question relates to the role that variant chain fatty acids play in the progression of ALD, and if we know what they play, then can we infer from that to what level we need to correct them to have an appropriate therapy. Well, the simple answer is if you don't have elevated levels of variant chain fatty acids, you don't develop ALD. How low do we need to go with the levels of variant chain fatty acids? That honestly I would not know. We will get some sort of answer from studies that we do in skin fibroblasts. We have a study that we hope to publish soon, in which we demonstrate the toxic role of very long chain fatty acids in ALD, and we have very clear and very sensitive outcome measurements, and we could titrate the concentration of very long chain fatty acids in that cell type to see if at a certain level the cells behave normal. Of course, that's the limitations. The major limitations are that this is a laboratory setup, so it's in skin fibroblasts from patients. But this would be a typical question that could come out of the clinic, because it directly relates to the efficacy of any very long chain fatty acid lowering therapeutic agent. The most important thing is that if we want to be effective, we need to lower the very long chain fatty acid levels in the target tissues, and that would be the brain, the spinal cord, the adrenal gland, and the testes. So what's my best scientific guess? I would opt for near normalization. That there are no more questions, then again I would like to thank you for your attention, and yeah, maybe we'll meet again in another webinar. Have a good day.